Hey Mustangs, in this video we're going to be taking a look at the first step of protein synthesis, which is transcription. So if you remember, there's transcription and translation. The best way to remember which one comes first is take both of those and put them in alphabetical order. So if you take the words themselves and put them in alphabetical order, you'd find they both start with trans, but then here you have a C for scription, and then translation has an L. So if you put them in alphabetical order, transcription always comes before translation. Now additionally, the word itself here, transcription, can help us understand what's going on. So the word script means to write. So in transcription, it's all about rewriting DNA instructions into mRNA instructions again, instead. So transcription and translation. Here we're looking at transcription first. So this video is all about transcription. All right. So first, in order to really understand transcription, we have to understand genes. So if we look at your DNA, most of your DNA is actually junk DNA. It has no instructions for building proteins or mRNA or RNA, and those sections are called junk DNA. The other sections of your DNA that actually do have instructions for making a protein are called genes. So a section of DNA that has instructions for making a protein is called a gene. Now, don't be fooled by the name junk DNA. It's called junk DNA before we really fully understood DNA and all of its parts and stuff. So a lot of the stuff that's junk DNA actually isn't junk. It's sections of DNA that are actually used in helping read a gene, to transcribe a gene, identify where genes are. So junk DNA isn't unimportant parts of your DNA. It was just called that because it doesn't have instructions for making proteins. And the ones that do are called genes. Now, the central dogma of biology is basically referring to protein synthesis. It's the idea that DNA has instructions for making proteins. So the DNA spells out with the A's, G's, T's, and C's how to build proteins, very specifically saying exactly what amino acids are supposed to be connected together. DNA is then rewritten into RNA, so into mRNA. Now here are the instructions. Those instructions say the exact same thing as this, just written in a different form. So rewritten the instructions into mRNA, and then from there, those instructions are read. So you read the instructions in the mRNA and it tells you how to build a protein. So that's known as the central dogma of biology here. So DNA has instructions, get rewritten into mRNA, the mRNA is read, and proteins are built. Very specific. So the DNA is very specific, saying exactly which amino acids to connect together, in what order to build your proteins. So here is a visual example of the central dogma. So we have DNA rewritten into mRNA, and then a protein is made as the amino acids connect together to build a protein. On paper, transcription, remember script is rewrite, it's really easy. So if we take a look here, if I transcribe this on paper, every single time you see a T, you write an A. Every single time you see a you write a U. Every single time you see a C, you write a G. And I like to tell my students good habits here. So after every 3, you draw a line. So then every single time you see a G, you write a C, A, U, line, G, U, G, line. So this is almost like making a copy of DNA, with the exception of the fact that every single time you see an A, it goes to a U instead, to a uracil. So that's how you transcribe on paper. Now, what's really happening in the cell is what you need to know. So there's initiation. Initiation is the start. So first, transcription factors bind on or near the ta-ta box of the promoter. RNA polymerase II binds with additional transcription factors, forming a transcription complex, and RNA polymerase begins to unwind the DNA. So let's take a look at what we're actually talking about here. So there is a promoter region. Here's the ta-ta box. Promoter region is basically the starting point. This is an example of junk DNA being used in order to identify where a gene is. Okay, so the gene would be over here, past this point. So promoters are always in front of the genes here. On the promoter, this is where transcription factors and RNA polymerase will assemble. RNA polymerase, don't confuse that with DNA polymerase. DNA polymerase builds DNA during DNA replication. RNA polymerase is going to be used during transcription to make mRNA instead. Okay, so this is the basics. There's a promoter region, transcription factors. All of this must be assembled. So the promoter region is basically a spot for all these necessary materials that are needed for transcription to occur to assemble in one place. And all of this needs to be assembled in order for transcription to occur. But it's a little bit more complicated than this. So key things here are the promoter, transcription factors, and the RNA polymerase. So the full picture with transcription adds a little bit more to what we saw in the previous slide. So in the previous slide, we focused on the promoter region, where this is where the RNA polymerase will bind, this is where transcription factors will bind. And there's a little bit more to it when you talk about eukaryotes. So in eukaryotes, there's a section of the DNA further past the promoter region called the enhancer, or sometimes even the silencer, depending on if it's going to turn on or turn off a gene. And with activators, these are molecules that must bind to it in order for a gene to actually start transcription. So here, these could actually be based on steroid hormones. So if you remember, steroid hormones can enter into a cell because they are hydrophobic. Once inside, they bind to a protein receptor, and this could be basically a steroid hormone bound to its protein receptor, and it can act as an activator in order to start transcription. Or you could have amino acid or protein hormones that can't enter a cell because they're hydrophilic. They bind to a surface protein and then start a signal transduction pathway that turns on activators. So in any case, you need these activators in many genes to bind to this enhancer region of the DNA. Once that happens, another protein, a DNA binding protein, will bind, and it bends the DNA over so that the enhancer, along with the activators, are over the promoter region. And then here you have several different proteins, including your transcription factors, that will all bind to the promoter region. Once this whole thing is assembled, then transcription can happen. So this is in eukaryotes. In prokaryotes, it's a lot more simple, and that's in another video. So prokaryotes, it's more simple than this, but in eukaryotes, there's more complex things that have to come together. So you have the enhancer with the activators, and these can be a result of steroid hormones or amino acid hormones or other factors. And then you have in purple, the transcription factors, and then this light-colored molecule here is the RNA polymerase. Now don't confuse RNA polymerase again with DNA polymerase. Make sure you know RNA polymerase 2 in this case is involved with transcription. In order for it to actually build the mRNA, take a look here, there's elongation, it's actually building the mRNA. So DNA polymerase unwinds the DNA, it adds nucleotides 5' to 3' direction still. So even RNA is made from the 5' to 3' direction, and then the mRNA peels away. So if we take a look here, in blue we have the DNA, in red we have the mRNA being built. So here's the 5' end of the mRNA, here's the 3' end, and we see them being built 5' to 3'. And then we have our polymerase and stuff here. So transcription will stop once it hits a termination sequence at the very end. And then there's RNA processing. So RNA processing is basically what happens in eukaryotes. Prokaryotes do not go through RNA processing. So after a mRNA has been made, the mRNA moves away, moves out into the nucleus. So it detaches from the DNA, moves out into the nucleus. The DNA actually zips back up. So the DNA is unchanged. It was just used to make a copy of the instructions into mRNA. The mRNA leaves, the DNA closes back up. That mRNA, before it leaves the nucleus, if it's a eukaryote, it must go through RNA processing. So it gets a 5' cap and a poly A tail. So here's the 5' cap and a poly A tail here. And then there's additional things. There's RNA splicing. So in order to make a mature mRNA that's going to leave the nucleus, you have your 5' cap, poly A tail, but then you have these sections called introns and sections called exons. Okay, so here the introns are actually going to be cut out and removed, and the exons will be spliced together by certain molecules called spliceosomes. So here is what would happen. The introns would be cut out by the spliceosomes, and the exons would be spliced together. So this would officially be a mature mRNA. It's not until the introns get cut out that you have a mature RNA here.